Good afternoon to those that may have already joined us. I'm just checking, can anyone out there hear me? Is the sound coming through? Afternoon, Clifford. Can you hear me? Is it coming through on the speaker? Great. Okay, we have about 2-3 minutes before we start, so we'll just sit and wait for a few moments, and we'll start at 1 o'clock sharp. Once again, if you're just joining us, we're going to start at 1 o'clock sharp, so be patient and we'll get going here in just a moment. Okay, good morning. It is 1 o'clock here in Michigan time, and we are going to begin our third session of live stream reviews through YouTube. This is Unit 3, Cellular Energetics. My name is Mark Eberhard. I'm a high school teacher in St. Clair High School, which is in St. Clair, Michigan. And we are going to jump right in because I have a very heavy agenda, lots to try to get through. First, a couple basics for your information. We have several expert AP Bio teachers joining us in the chat room today. They will be monitoring the chat room and answering your questions throughout the session, so please feel free to put your questions in the chat room as we go along, and these excellent AP Bio teachers will be glad to respond. Below this video presentation, you will find links. There are links for future presentations that are coming up, which are coming up the next 3 or 4 weekends. There's a schedule of what sessions will be happening when. There'll be a link to this slideshow. There's also a question submission form. So if you have particular questions about a particular unit, you can go to that form and submit your questions ahead of time. We ask that you try to do that about a week in advance so the presenters can build that into their program. This is a review, So keep in mind that we're going to try to review the key concepts associated with cellular energetics. We only have about an hour today. Typically, I know I spend about 3 weeks on this unit alone, so we can certainly not cover everything we would do in 3 weeks in an hour session. So the assumption is that you have already gone through that 3-week learning period, and we are going to try to highlight some of the key points and concepts that you might see in the national exam. We're also going to, at the end, explore how this might be incorporated into this year's exam. And we hope to support, not replace, your own review materials for cellular energetics. So with that said, let's go ahead and jump right in. In Unit 3, there are actually 7 topics, and you'll notice I've actually not started with topic 3.1. I've decided to start with topic 3.4, which is cellular energy. From my own experience, I think it's better to have an understanding of what's going on with energy before we jump into the other topics. So the learning objective here under 3.4 is describe the role of energy in living organisms. Why do we need energy? What's its purpose? We know that we eat on a regular basis, we consume food on a regular basis, and we get 2 things from our food. We get energy and we get matter. So what's the point of the energy? Well, if you ask a typical freshman class, they're going to say to live. Well, in AP Bio, we want to be able to go a little beyond to live. So it comes into this idea of laws of thermodynamics. The first law of thermodynamics says energy can be transferred and or transformed, but it cannot be created or destroyed. We have to have the ability to transform energy into different various forms that we need to carry out life's functions. For example, in this photo here, there are 2 basic energy transformations. We eat food and we convert that chemical energy into kinetic energy. Energy that's in action that allows us to do things like muscle movements. Another key process is photosynthesis, where we're going to take light energy and transform that into chemical energy to make it accessible to all the living things. Well, no matter what the energy transformation is, excuse me, during every energy transformation, some energy is going to be converted into thermal energy or released as heat. And when energy is released as heat, it's not lost, but it's no longer available to living organisms for doing work. So that means we have to find energy to replace that, and that's where the sun comes in. So long as the sun is providing a new source of energy on a daily basis, we can get away with this heat loss. We say that energy has a one-way flow. You may recall that matter is cycled, used over and over again, but energy has a one-way flow. Energy comes in from the sun, and as far as the living system is concerned, it's released in the form of heat. This leads to the second law of thermodynamics, which says that the energy transfer, during an energy transfer, the potential energy of the final state is going to be less than the potential energy of the initial state. This is because, if you reference our picture, a certain portion of energy is going to be lost as heat. This also could be stated in other words, every energy transformation increases the entropy of the universe. Entropy is disorder, disorganization, chaos. Nature favors disorganization. And because it's more simplistic, it requires fewer chemical bonds, the less investment of energy, things move towards some more simplistic state. They move towards this lower energy state. Well, the reason you need to provide yourself with food every day is your body is a highly organized structure. All the molecules that form your body are very organized, highly organized. Now, you personally may be completely disorganized, but your body molecularly is highly organized, and it requires the constant input of energy to maintain that organization because nature is going to naturally push you towards a state of entropy, towards a state of disorganization. So through most of our life, we will need to consume food on a regular basis in greater quantities than our body's going to expend it. Over time, we become a little less efficient and a little less efficient, and eventually we get to a point where our body is no longer efficient enough and we simply can't supply the energy we need to maintain the organization. And at this point, we call this death. Now, in terms of energy flow, we call the overall process metabolism. Metabolism is the sum of all the reactions in the body that involve the transfer of matter and energy. There are two basic pathways that we follow. There is the catabolic pathway and the anabolic pathway. In our diagram here, the first picture is dealing with an anabolic pathway. Anabolic pathways take small molecules. If we go back to chemistry of life, we can call these monomers. And we're going to take these individual monomers and we want to string them together into a more complex molecule called a polymer. Well, in order to take these monomers and convert them into the polymer, we have to form chemical bonds. We're going to require more bonds, more structure, more organization. To do that, that requires the input, a net input of energy. So in anabolic reactions, we are going to synthesize, we're going to build complex molecules, which requires a net input of energy because we're going to need to create chemical bonds. The opposite of that is a catabolic reaction. In a catabolic reaction, we're doing just the opposite. We're taking the polymer, the more complex molecule, and we are going to break those bonds to recreate the monomers. In this case, we're going from something organized, more chemical bonds, more complex, and we're going to convert it into something that is less complex and requires fewer chemical bonds. Well, since you require fewer chemical bonds, the energy that was initially invested in the bonds in the reactant no longer is needed by the product, so that energy is now available to do work. And so we have a net release of energy. So anabolic, we're going from simple, fewer bonds to more complex, more organized, more bonds. That involves the investment, net investment of energy. In a catabolic reaction, we're doing the opposite. We're going from something like a protein to the monomers, amino acids, fewer bonds, less organization. Therefore, there's going to be energy left over, which can be invested in other work in the body. Another type of terminology that's used for this are the words endergonic and exergonic. It comes to another category called free energy. Free energy is the energy in the system that's available to do work. And so in an exergonic reaction, which is equivalent to a catabolic reaction, we are going to be breaking down complex molecules into simpler molecules. That's going to require less organization. Fewer bonds are going to be required. Therefore, there's going to be free energy left over to do work. Because we're going from complex to less complex, fewer bonds are required, thus less energy is required. Thus is going to be, in a sense, leftover energy available to do work that can be invested elsewhere in the body. So we can somewhat use the words catabolic and exergonic interchangeably. Endergonic is equivalent to anabolic because we are going to do the opposite. We're going to synthesize more complex molecules. We're going to increase the level of organization. More bonds are going to be formed, and we have a net input of energy in order to make this happen. So, one set of reactions is ending up with leftover energy that's no longer needed. Another set of reactions actually requires the investment of energy. So what the living organisms have learned to do is to connect those two reactions. And when we connect these two reactions, we call this energetic coupling. This is let me back up a step first before we go to coupling. This is just a diagram of an exergonic and endergonic reaction. And it visually shows you pretty well what's happening. Here we can see that in an exergonic reaction, we're starting with something that's got more energy invested in it, more complex molecules. We're converting those into a product that is less complex and requires less energy. Thus, there's excess energy being released, a net release of energy, which creates a delta, what's something called a delta G, is a measurement of free energy. This net delta G is going to be less than zero or negative. This makes this reaction spontaneous. And in the opposite picture, endergonic, you can see it's just the opposite. We're taking simpler molecules that require fewer bonds and less energy and converting them into more complex molecules which require a higher level of energy. Thus, in this case, we're investing energy. And that's going to create a positive delta G. So again, what living organisms have learned to do is to connect these two reactions. We call this energy coupling. In energy coupling, we're going to use the reactions that release energy, the exergonic reactions or catabolic reactions, to drive the reactions that require energy, the endergonic or anabolic reactions. So over here, we have energy from catabolism or exergonic releasing reactions. That energy, we want to transfer it to the endergonic reactions, but we need a go-between. We need something to help us move the energy from one set of reactions to the other, and that's where ATP comes in. So the energy from the catabolic reactions or exergonic releasing reactions is going to be invested in phosphorylating, adding a phosphate to this molecule called ADP, adenosine diphosphate, to form ATP, which is a more highly energized molecule. That ATP is then going to be hydrolyzed, adding water, and that is going to then release the phosphate back off of the ATP, which is going to allow you to release the energy to the endergonic or anabolic reactions, and thus the energy is transferred. So the body, for every endergonic reaction the body has, it needs to have an exergonic reaction to provide the energy. And so exergonic reactions provide the energy to fuel the endergonic reactions our bodies use. And these are connected. What couples these together is the production of ATP. ATP is the molecule our body uses for transferring energy. It's the energy currency of the body. ATP itself is a very unstable, short-lived molecule. It actually can only be used in the cell that produces. It's not transferable from one cell to another. Thus, if you want to store the energy and be able to move it around, you have to put it into a more stable molecule like an organic molecule, which we'll get to. But the nice thing about ATP is it stores a small, safe amount of energy that's usable to carry out a typical reaction. When you remove the phosphate and release the energy, you're releasing energy in safe doses. In other words, you're not releasing energy in such large quantities that you're going to damage the cell. Whereas if you were trying to pull the glucose molecule together all apart all at once, too much energy too quick, you're going to damage the cell. Not so with ATP. So, bottom line, in terms of cell energy, our bodies need energy on a daily basis in order to maintain organization. And life is about taking polymers from our environment, breaking them back down into monomers, and then using those monomers to create the polymers our body needs. And in the process, we're going to store and release energy. So maybe last night you ate a piece of chicken, you ate a chicken protein. Your body is going to catabolize that protein, digest it, break it down into its amino acids. Your body is going to absorb those amino acids and deliver them to your cells, and your cells are then going to invest energy in rearranging those amino acids in, say, to a human protein. So some of this transfer is transferring matter, which I just described, and then others, of course, is transferring energy. So that brings us to topic 3.1. I'm moving quickly, so if I'm going too fast, let me know. The topic 3.1 gets into enzyme structure. So these catabolic and anabolic reactions that we're describing, these reactions that we need to sustain life, on their own are too slow. They just don't happen very efficiently. They don't happen very quickly. So for example, if I take some type of food source, let's say, for example, I have in my hand here, I happen to have 2 raspberries. Now there's a slight delay, but you're going to see 2 raspberries come up. And if I just set these 2 raspberries on my table, will they eventually break down? Will they eventually decompose? Of course they will. But that may take weeks, that might take months in order for that to happen. And your body can't wait days, weeks, months for the chemical bonds to break down and the energy to be released or the energy to be stored. So we need a way to speed up the process. We need a way to make those reactions happen at a more efficient rate. And that's where these molecules called enzymes come into play. So let's look first at the structure of the enzyme. So it says, describe the properties of an enzyme. Well, enzymes, most of them are protein-based. There are some enzymes that are RNA-based, but we're going to focus on the majority of enzymes that are protein-based. Therefore, shape determines function. Remember, in proteins, very important, shape is extremely important. Shape determines the function. And the most important part of the protein of an enzyme is a region here called the active site. The active site is the most critical part of the molecule. The shape really matters in the active site. The shape is designed to fit a specific substrate. A substrate is the general name for any type of molecule that interacts with the enzyme. In the earlier days, we used to have what was called the lock and key model. The idea was that the substrate was like a key that fit into a lock like the enzyme, and that each enzyme would only have one substrate, one key. You know, your key that opens the front door to your house doesn't open everybody else's front door because it has a unique shape. Since then, we've revised that model into what's called the induced fit model. We now realize that the active site actually has a little bit of chemical give, and it can actually mold a little bit around the substrate, which opens up the idea that some enzymes may actually be able to interact with a couple different substrates. Generally, they'll be very closely related in the same family, but the newer theory is the idea that there is a little give in that active site, and it has some adaptability during the reaction. Here is a more detailed picture. So shape is definitely one of the factors. Right here, this is the active site of the enzyme. This is more of a schematic drawing. This is the active site of the enzyme, and the green substrate is sitting in that active site. And if you look at that, right where I just put the yellow circle, there is an attraction by charge. The plus region of the substrate is attracted to the negative region of the active site. Over here, where I'm placing the fuchsia-colored circle, you see there's a positive region in the active site, which is attracting a negative region of the substrate. And of course, there are also some hydrogen interactions going on here. So not only is shape important to determining what fits into the active site, but charge also plays an important role in what fits into the active site. These other alternatives, B, C, D, and E, right where they have the arrow, there's a reason why that substrate does not fit that active site. In B, this extension piece is going to interfere with the bonding of these hydrogen bonds that are forming over here. This is not going to allow this to set into the active site deep enough to have that interaction. Something like molecule D, you can see you have a negative, got the right shape, but you got a negative region there instead of a positive region right here. And therefore, if you try to put a negative against a negative, it's going to be repelled. So substrates have in active sites, there's a good deal of specificity that is there. Enzymes do very specific jobs. Enzymes that are catabolic and break things down, that's what they do. And typically they only work with particular substrates. A whole different set of enzymes are needed to carry out anabolic reactions. So this is an overall picture of basic enzyme function. So here you can see you have the active site and the substrate. The substrate binds to the active site based on shape and charge. When it's in that active site, in this particular case, we're going to stress the bond. We're going to put a lot of stress on the bond, and we're going to cause this bond to break. And that's going to cause the separation of these two subunits or two monomers, and the new product is produced. In this particular case, we're catalyzing the breakdown of substrate into two individual products. So what is the enzyme doing by doing this? The enzyme is going to do what we call catalysis. It's going to catalyze the reaction, which catalyze means it's going to speed the reaction up. It's going to make it go faster. So how does an enzyme actually make a reaction go faster? Well, here's a very standard reaction. This is, as you might recognize, cell respiration. We're going to take glucose and oxygen, which are our reactants, And we're going to convert those into products of carbon dioxide and water. The red line here represents the reaction without an enzyme. Every reaction requires what's called activation energy. Activation energy is the energy it takes to get the reaction started. Again, if I just leave this raspberry sit on my table and wait for it to break down, that's going to take a long time, because you have to get the proper activation energy. Same idea if I took a handful of amino acids and threw them on the table and waited for them to spontaneously form a protein, I'm going to be sitting here a long time. So the activation is what active energy has to be reached. Once you reach the activation energy, then the reaction can take off and you can produce your products. Well, the red line represents what happens when you don't have an enzyme. You can see it requires a much higher level of activation energy. What an enzyme does is an enzyme reduces the amount of activation energy, which is the blue line, that you need to get the reaction started. By lowering the activation energy, the reaction can take place much faster than it would if you had to achieve the original activation energy. And the nice thing about this, so here's our activation energy. You can see we've cut that almost into a third in this particular case. The nice thing about it is whether you use an enzyme or not, the overall amount of energy being released doesn't change. It doesn't change. You still get the same output. You still get the same output. Okay, so enzymes lower the activation energy. Well, how do they do that? How does an enzyme lower the activation energy? We'll talk about a couple ways, if not all of the ways, but one of the ways an enzyme lowers the activation energy, let's go to a synthesis reaction, an anabolic reaction, where we want to make a product by combining molecules. Well, if you are going to combine molecules, they have to come in contact. And they not only have to come in contact, they have to come in contact in the correct orientation. The right parts of the molecule have to make contact with the other correct parts of the other molecule in order to form the bonds that are necessary to form the compound. Well, a lot of these things happen in an aqueous environment in the body, water-based environment. And if you're just going to randomly wait for these two molecules to bump into each other and then hope that when they do bump into each other, they bump into each other in an orientation that allows them to bond together, that's going to take some time. Whereas if you have an enzyme is designed to pick up the necessary components, and not only pick up the necessary components, here a red piece and a yellow piece, but once they're in the active site, we're going to actually put them in the correct orientation, which is going to favor their ability to make the chemical bonds that are necessary to form our orange product. So, two ways that enzymes lower the activation energy as far as the synthesis reaction is concerned is they bring molecules together in close contact, and they also put them in the correct orientation, which favors the proper formation of bonds to create the necessary product. On the other hand, if you look at something like a decomposition or catabolic reaction, here we have the sugar sucrose, this yellow sugar here, which is bonding to this purple enzyme. What's going to happen in this particular case, as it says under number 3, is the binding of the substrate in the enzyme places stress on the glucose-fructose bond. This enzyme is actually going to apply some torque, some stress on that bond, which is going to make it very difficult for sucrose to maintain that bond. That bond will be much more easily broken, and we will produce the two monomer products of glucose and fructose. So enzymes can catalyze reactions by being able to bring molecules together in the right orientation, in the favorable conditions that are going to best promote the reaction to move forward. And thus, we can make these things take place a lot more efficiently. Now, because a lot of enzymes are proteins, they are susceptible to environmental factors. So to explain how the changes to the structure of an enzyme may affect its function. Now again, these are proteins, and remember, one of the things that can happen to proteins, if you don't have the right conditions, proteins can go from its three-dimensional shape, its tertiary structure, which we see here on the left, and it can quickly unravel and become unfolded, and now you've changed the shape of the protein. And if you change the shape of the protein, you are highly likely to change its function. And in this case, if it's an enzyme, this protein probably is no longer capable of catalyzing the reaction. So what might do that? Well, two of the key factors that can affect protein stability and enzyme stability is temperature and pH. So on this particular graph, this is a pretty basic graph, but on this particular graph, if you are in the optimal conditions, the good conditions, whether it be pH or temperature, the protein maintains its three-dimensional shape, and if it maintains its three-dimensional shape, it can catalyze the reaction. As you get further away from the good conditions, you get into more stressful conditions, that's going to start to affect the shape of the protein. And you're going to start losing some enzyme function, and we're going to start to see the reaction rate drop. And if you get too far away from the good conditions, from the optimal conditions, you're going to cause the complete denaturation of the enzyme. And at this point, you're going to shut down the reaction. The enzyme is going to no longer function. So the environment is very critical for proper enzyme function. So let's take a look at each one just briefly. Here we're looking at pH. pH can disrupt the hydrogen bonds that provide enzyme structure. So remember, if you're getting into a very acidic pH, you have a lot of free hydrogen ions that are in the environment. Those positive hydrogen ions can interfere with the bonding within the enzyme's protein structure, and it can cause a change in that structure, which could cause a change in the active site, which could cause the enzyme not to function. Vice versa, if it's a very basic solution, you've got a lot of negative hydroxide ions, which also can interfere with the hydrogen bonding and the ability of the protein to maintain its structure. The other thing that we see in this graph is that not every enzyme has the same preferred pH. Not every enzyme has the same optimal pH, even in our own bodies. Here on the left, you see the black curve. That represents the enzyme pepsin, which is in our stomachs, and that prefers a pH of 2. Whereas if you go to another enzyme called trypsin, which, by the way, both these enzymes break down protein, in the intestinal environment, it works best at a pH of 8. So these two enzymes are not going to function in each other's environment. Put trypsin inside the stomach, it's going to do nothing. Put pepsin inside the intestine, it's not going to function. And notice if you move out a half a pH unit, a whole pH unit out from the optimum, there's a big drop in the curve. We're starting to stress those enzymes. They're starting to lose shape. They're starting not to function. And in both these cases, if you move out somewhere between 1.5 to 2 pH units, you're going to totally denature the enzyme, whether you go too acidic or too basic, and it's no longer going to function. And you're going to shut down the reactions. Temperature, you see a similar curve, though a little bit different. Again, different enzymes can have different optimal temperatures. Most of the enzymes in the human body, the black curve, prefer temperature right around 37 degrees Celsius, which is body temp. No surprise. There's a group of bacteria that you can find in the vents, like in places like in Yellowstone, where they have very hot, steamy environments. These thermobacteria prefer an environment of 75 degrees Celsius. But you can see they both have similar shapes. On one end, if the temperature drops below the optimum, we're not so much in danger of denaturing, but what happens as you cool things down, of course, is molecules slow down. And as molecules slow down, you're going to reduce the number of collisions, contact between the enzyme and the substrate, and we're going to see a considerable drop-off on the reaction rate. Vice versa, if you increase the temperature, of course, the molecules are going to increase activity. Collision rates between enzymes and substrate will increase, and at least for a little bit, you'll see an increase in the reaction rate. But notice in both cases, if you get about 12 degrees, 15 degrees away from the optimum, boom, that reaction just completely shuts down. Because at some point, the temperature gets too high, the Protein, again, cannot maintain its tertiary structure. It is not able to maintain that three-dimensional shape. Again, you have denaturation, you have a change in protein structure, and the enzyme can't function. Another key variable in the environment can be either the substrate concentration or the enzyme concentration. This particular graph looks at substrate concentration. And yes, to a point, if you increase substrate, you're going to increase the reaction rate. The more substrate there is, the more substrate that the enzyme can either break down or synthesize. Up to a point. At some point, it will level out. Why will it level out? Because at some point you're going to be at maximum production. Every enzyme is engaged, every enzyme is catalyzing the reaction, and there just isn't any more enzyme available to make the reaction go any faster. So if you've got, say, 100 boxes to move and you can carry one box at a time, and you've got 10 people to move those boxes, that's going to take you 10 trips. If you increase the number of workers, which is like increasing the enzyme, you now have 20 workers, you can do that in 5 trips. If you increase that to 50 workers, you can do that in 2 trips. If you increase that into 100 workers, you're going to do it in 1 trip. But after 100 workers, it doesn't matter if you have 150, 200, 300, 400 workers, it's still going to be 1 trip. It's going to level out. And the same thing can be said with the substrate concentration. You hit a point what's called saturation. So those are environmental influences, key environmental influences: pH, temperature, enzyme concentration, substrate concentration. In lab and class, you may have also looked at factors such as salinity. Those can all influence the rate of the reaction. In addition, the cell itself wants some ability to control enzyme activity. Your body produces thousands of different enzymes to carry out thousands of different of reactions. But you don't need every enzyme to carry out every reaction 24/7. So the body does not want to have enzymes active, and the body does not want to produce enzymes if they're not needed. This is energy inefficient, this is material inefficient. We need to have a way to turn enzymes on and off and only have them on when we need them. One of the ways that enzymes can be regulated is through what's called competitive and non-competitive inhibition. Here in this cartoon, we see an example of competitive inhibition. The yellow structure, of course, is the enzyme, and the blue structure is the normal substrate. Normally, that blue substrate would fit into the active site of the yellow enzyme, and let's say in this case it would be a catabolic reaction, and we would split that blue molecule into a couple pieces. Well, one of those pieces was the green molecule. That was 1/2 of the breakdown. Well, that green molecule, if it accumulates because the body's not using it, you've got too much of it, that green molecule can compete for the active site. And when the green molecule binds into the active site, competes for the active site, it's blocking the active site, which is shutting down the breakdown of the blue substrate. Now, typically this is reversible. It's a numbers game. As the body consumes and uses these green molecules, you're going to pull that green molecule out of the active site, which opens it back up for the blue substrate, and we turn the enzyme back on, and the reaction continues until we accumulate too many green molecules, which will then come back and outcompete the blue molecule for the active site and shut the enzyme back off. Once the green material is used up, active sites open back up, turn it back on, and I think you see the repetition that happens. On a side note, this is what a lot of toxins do. A lot of poisons, something like a snake venom. These toxins compete for the active site. Unfortunately, sometimes these toxins, though, are what we call irreversible competitive inhibitors, that once they bind, they're there permanently. And in some cases with certain, for example, snake venoms, that can cause an irreversible inhibition, which causes muscle paralysis. The other side of this is non-competitive inhibition. Non-competitive inhibition is because you're not competing for the active site. They're going to use a totally separate binding site for the reaction to take place. There is another binding site on the enzyme called the allosteric site. In this particular picture, you can see it's right here. Here is the allosteric site compared to over here where there is the active site. And so in this case, the inhibitor is designed to fit the shape of the allosteric site, not the active site. Some allosteric interactions inhibit, they turn the reaction off. So this molecule, again, which oftentimes can be the end product of the reaction, if you got too much of it, will come in and bind to the allosteric site, which you can see changes the shape of the active site, preventing the binding of the substrate, which at least tentatively turns the reaction off. We'll see also that you can go the other way. The enzyme can be in an off mode normally, and the allosteric effector, which in this case we call an activator, can bind to the site, which actually rearranges the active site to become active, and now we can break the substrate down. So in allosteric regulation, some work as inhibitors, where they change the active site and turn it off, and some molecules work as activators, where they actually change the shape of the active site to turn it on. Here they are side by side. Competitive inhibition, you can see the blue molecule is blocking the active site and preventing the substrate from binding. In most cases, that blue molecule will eventually be removed from the active site and the substrate will take it over and the reaction will continue. And compare that to the one on the right, you see that yellowish molecule binding to a separate site called the allosteric site, which is slightly altering the shape of the active site and preventing the reaction with the substrate. Both of these are ways that the body can turn these enzymes on or off so that they're only being used when needed. A lot of times enzymes are also arranged into what's called feedback inhibition. It's a pathway. A lot of enzymes work in a pathway. This being the pathway, here we have the substrate, the green substrate triangle, reacting with enzyme 1, which converts it to a purple rectangle. The purple rectangle reacts with enzyme 2, which turns it into this blue odd-shaped piece. This blue odd-shaped piece combines with enzyme 3, which then produces the final desirable product, the orange square. Pathways have a lot of advantages. I'm not going to go into a huge discussion on pathways right now, but pathways give you multiple points to regulate the process. It gives you multiple points where you can increase production, decrease production, turn production on, turn production off. And a lot of times what happens is the end product ends up being the feedback. Again, if you're producing more product than you can use, that end product is going to travel to the original enzyme, enzyme 1. It's going to bind to an allosteric site, and it's going to change the active site, which turns off the reaction. That enzyme will stay turned off until the end product gets used up, and that is removed from the allosteric site, and then the enzyme will regain its original shape and turn the reaction back on. So these are all about energy efficiency, material efficiency, allowing enzymes to only be on when we actually need them. So enzymes allow reactions to happen at an efficient rate at which life can exist. The active site shape matters. That is the key part of the molecule that drives the reaction. Environmental conditions can negatively impact enzyme function, such as temperature, pH, and concentration. And the body has several mechanisms, both competitive inhibition, non-competitive inhibition, allosteric activators, and feedback pathways, all to help regulate and control when enzymes are turned on and off. So that takes us to the two big reactions under cell energetics, photosynthesis and cell respiration. We're going to start first with photosynthesis. This is where it starts, the beginning of it all. Photosynthesis is the ultimate endergonic anabolic reaction. So most of us should already know this equation, that in general photosynthesis we're taking carbon dioxide and water, two simple molecules with very few bonds, and we're going to convert them into sugar, which is a more complex model involving more chemical bonds, which means it takes the investment of energy, which is ultimately going to come from the sun. And as a result of that reaction, we're going to produce the waste gas oxygen. Now, both cell respiration and photosynthesis involve what's called oxidation-reduction reactions. Oxidation-reduction reactions are what drive both photosynthesis and cell respiration. So if we look at this reaction of photosynthesis, you can see that we are going to oxidize the water. That's the pinkish red arrow. We're going to take water and oxidize it. When we oxidize, it means we're going to remove electrons. We're going to lose electrons. And those electrons often take with them the energy and are also accompanied by hydrogen. So we're going to take water, we're going to oxidize it into oxygen. Now, if you oxidize something, you have to have a reduction. You can't oxidize without reducing, you can't reduce without oxidizing. And so in this equation, what's going to happen is the carbon dioxide is going to receive those electrons and energy, and in biological systems often follow the hydrogen. And so the carbon dioxide is going to pick up the electrons and the hydrogen that you oxidized from the water, and it's going to use that to form glucose, sugar, C6H12O6. So one of the ways to remember it, there's a saying here called Leo goes grr, you may have heard of. Leo stands for lose electrons oxidation, and grr stands for gain electrons reduction. Sometimes that confuses students. Gain, reduce, what do you mean? We're gaining, but we're reducing. Remember, electrons have negative charges. So when you gain electrons, you're reducing the overall charge of the molecule. That's why we call it a reduction. So keep that in mind as we go. Another saying that some teachers like to use is oil rig. Oil rig is another way to remember this. Oil for oxidation is losing electrons, and rig for reduction is gaining electrons. Whichever one works for you. Leo goes grr, oil rig. Just keep in mind oxidation, we're losing electrons. Wherever the electrons go, so goes the energy. And in biological systems also usually follow the hydrogens. Reduction, just the opposite. You're gaining electrons, which means you're gaining energy. And in most biological systems, that means you're also going to attract hydrogen. Now recall, all of this takes place in a cell organelle called the chloroplast. We're going to focus on a couple areas in the chloroplast today. We're going to look at the thylakoid and thylakoid space. That's these green stacks. This is where the first phase, the light reactions, are going to occur. And then around these green stacks, hard to see in the picture, there's a dense fluid called the stroma. And it's in the stroma that we're going to carry out the second phase called the Calvin cycle. Just as a quick side note, because this is listed under 3.5 in the curriculum, that we believe chloroplasts, there's a lot of evidence to support that chloroplasts came about through a process called endosymbiosis. Now, a lot of teachers usually will cover this when they cover evolution. Some of you may not have gotten to Unit 7 yet and may not have covered evolution yet, but I'm going to keep this really simple. The concept is, the evidence is, that eukaryotes, early eukaryotes, engulfed these photosynthetic bacterium, and over time, these photosynthetic bacterium sort of became permanent residents. We formed a symbiotic relationship, and those photosynthetic bacteria basically gave rise to the chloroplast. Some of the evidence is chloroplasts have their own DNA. They have their own ribosomes. They can reproduce on their own without the rest of the cell reproducing. They have membrane structures similar to prokaryotes. But for our purposes, endosymbiosis is the theory that chloroplast and mitochondria were taken in by early eukaryotes and formed symbiotic relationships that gave rise to these two organelles. So So let's take a look just at a broad overview first. As we go into this in cell respiration, let me be very clear. You are not expected to memorize names of enzymes. You are not expected to memorize steps, specific steps. You will never be asked to draw any of these diagrams we're about to go through. Really, what you need to understand is what are the inputs and the outputs? What's going in, what's happening, what's coming out, and how are they connected? So don't worry about having to redraw any of these steps. Don't worry about having to memorize names of steps and names of molecules. That you're going to see is beyond the scope of the AP exam. So in a general overview, what's happening in photosynthesis is during the light reactions, which occur in the thylakoid, light is going to be absorbed, water is going to be oxidized, producing oxygen, and that is going to allow us to convert light energy into chemical energy in the form of ATP and something called NADPH. That energy is going to be used by the Calvin cycle to take CO2 and reduce it into organic molecules like sugar, and then regenerate the necessary NADP plus and ADP that is needed to make ATP and NADPH. And so if you can understand that much, you've got the main idea. The light reactions are all about taking light energy and converting it into two chemical forms, ATP and NADPH. That ATP and NADPH is then going to be transferred to the Calvin cycle, where that energy will be invested in reducing carbon dioxide into organic molecules. And in the process, we regenerate the carrier molecules of NADP plus and ADP so that we can keep repeating the process over and over again. So explain, how do we actually capture light? So we're going to move through this fairly quickly. Again, don't worry about memorizing specific names and structures. In the light reactions, there are photosystems. There's photosystem 1 and photosystem 2. And the job of the photosystem is to absorb the sunlight using chlorophyll and excite electrons. Now, the only crazy thing about this is the naming. Photosystem 2 actually comes first, and photosystem 1 actually comes second in the process. Why? Because they're named in the order in which they were discovered. And science sometimes is pretty stubborn about changing names. So, a little confusing, but photosystem 2 comes first, and it's named 2 because it was discovered second, and photosystem 1 comes second. Don't get too hung up on that, though. So what's happening here is we are going to absorb sunlight, right where I just put that blue circle. Sunlight is being absorbed by those green structures, which is the chlorophyll. That energy is transferred from the chlorophyll to this reaction center, where it says P60 or P680. And we're going to take a set of electrons and we're going to excite them. We're going to move them to a higher energy level. And that's the whole point of a photosystem, to take the light energy from the sun, transfer it to the electrons, and get them excited. We've just gone from an energy transfer. We've just transferred the energy from light to a chemical form in the form of electrons. Unfortunately, though, the electrons are not directly usable by plants and animals, so we're not done with the process yet. So we have these excited electrons. Well, now that we excited those electrons, we have to replace them with new electrons. That's where water comes in. Water is going to enter the photosystem. Well, water is going to come near the photosystem. It's going to be oxidized. And you can see it splits into several components. It's going to split into oxygen, which is going to be released as a waste gas. It's going to produce hydrogens, which are going to accumulate. And then it's going to release electrons to replace the electrons you just got excited. So water is the source of new electrons to continually replace the electrons that are getting excited and moved on. So by the time we get to the end of the first phase, photosystem 2, we have 2 excited electrons up here. Photosystem 2 is going to be connected to photosystem 1 through what's called an electron transport chain. This is the electron transport chain. And so these excited electrons are going to get passed from molecule to molecule as it moves down the electron transport chain. We show it in a downward direction because the electron is releasing energy as it travels down the electron transport chain. Ultimately, we're going to see that that energy is used to produce ATP. I'm going to come back to that in a minute as to how the energy from the electrons is converted into ATP. That electron anyway gets passed from photosystem 2 down an electron transport chain over to photosystem 1. That electron, as it goes from photosystem 2, right here where I've got the red thing, red dot, it comes down the electron transport chain, and it comes to the second photosystem, photosystem 1. And those electrons are going to get reboosted. Light is being absorbed by this photosystem as well, and that's going to reboost these electrons back up to a higher energy state. And these electrons are going to travel down another smaller electron transport chain where they will be accepted by what's called an electron carrier, NADP, to form NADPH. There's the reduction. We are going to reduce NADP into NADPH. So we're oxidizing water to get the electrons so that we can energize them to transfer the energy from light to chemical. And then we're going to reduce those electrons into a temporary carrier molecule called NADPH. That's just holding on to that energy in those electrons for the time being. Over here is the ATP. Let's take a look at how do you get that ATP. What I'm about to describe actually happens in both the chloroplast during photosynthesis and happens in the mitochondria during cell respiration. So now we're taking a look at that green membrane magnified. This is one of the thylakoid membranes magnified. Built into the membrane is the photosystem, the electron transport chain, and the other photosystem. So here's photosystem 2, here's the electron transport chain, here's photosystem 1. This is the thylakoid membrane. In here is the thylakoid space, and out here is that dense fluid called the stroma. So what's happening as the electron gets passed from molecule to molecule is it's releasing some of its energy, and that energy is being used by protein pumps to push hydrogen across the membrane into the thylakoid space. And this is going to build up a large proton gradient, a large collection of hydrogen. The key is the hydrogen is not permeable to the membrane. Once the hydrogen gets inside the thylakoid space, it's going to accumulate and it can't get back out. This builds up both a concentration gradient, where you have more hydrogen inside the thylakoid than outside, so it would like to follow the gradient and go from high to low concentration. You're also building up an electrical gradient, where the inside in relation to the outside is much more positive than the outside environment. So you've got several forces. You've got this concentration gradient wanting to push the hydrogen back across the membrane. You have this electrochemical gradient wanting to push the hydrogen back across the membrane. And so we're going to take advantage of that. We're going to take advantage of that. So just realize where I've put the blue circle, that's the electron transport chain. As the electrons are moving downhill, they're releasing energy, which is allowing some of the proteins to pump hydrogen into the thylakoid space. So now that we've got this buildup of hydrogen, we've got this huge buildup of potential energy, we need to tap into it. And so that's going to take place right here with this specialized protein called ATP synthase, which is going to provide a channel through which the hydrogens can cross back through the membrane into the stroma. And as these hydrogens rapidly move through this ATP synthase, they are going to provide the energy necessary to convert ADP and an inorganic phosphate into the highly energized molecule ATP. And so it's the buildup of the hydrogen gradient, it's the buildup of the proton gradient that's going to provide the energy necessary to phosphorylate ADP and form ATP. And so when we are done with the light reactions, some of the energy that was in the electrons has been invested in the formation of ATP, and some of the energy that was in the electrons is being temporarily held on to by this electron carrier called NADPH. And ATP and NADPH are going to both carry that energy to the second stage, which is the Calvin cycle. Now this is the Calvin cycle. This is beyond the scope. You do not need to know this diagram. You do not need to memorize anything about this diagram. But this is the key second step. What's going to happen in the Calvin cycle is we're going to take in carbon dioxide. We're going to use the energy from ATP and NADPH. All this ATP, all this NADPH is coming from the light reactions. We're going to basically oxidize the ATP and the NADPH so we can reduce carbon dioxide into this molecule called G3P. G3P then can be used to build multiple different types of organic molecules. 2 G3Ps is used to build glucose. But the plant can use that G3P to not only build glucose, but to build other carbohydrates, to build other proteins, to build other types of lipids in the process. So as far as the Calvin cycle, understand what's happening in the Calvin cycle is we're taking in CO2, we're using the energy and electrons and hydrogen from the ATP and NADPH to create this intermediate product called G3P, which in many cases is used to form glucose. Here's a good summary picture. If you can follow this summary picture as far as the steps, you'll have a pretty good idea of what you need to understand about photosynthesis. So in the light reactions, these are carried out in the thylakoid membrane. They convert light energy into 2 chemical forms, ATP and NADPH, which is the whole point of photosynthesis to begin with, is to convert light energy into chemical energy. And then we're going to split water and produce oxygen. During the Calvin cycle, which takes place in the stroma, we're going to use that energy to form CO2 into sugar, G3P, and to regenerate the ADP and NADP necessary for the light reactions. Why are we ultimately putting into organic molecules? Because in organic molecules, those are covalent bonds. They're much more stable. The energy now is transferable. The plant can't transfer its ATP to you. The plant can't transfer its NADPH to you. But the plant can transfer organic molecules. And that's the more stable form of energy. So the light reactions put the light energy into a fairly unstable chemical form, and the Calvin cycle, in a sense, puts these unstable chemical forms into a much more stable chemical form that now can be transferred between cells and be transferred between organisms. Again, the exclusion statement is that you do not need to know the steps of the Calvin cycle, the structures of the molecules, or the names of the enzymes. Outside of you should know ATP synthase. Okay, as I'm looking at the clock, I'm running slightly behind. This will take probably about 12 more minutes for those that are able to stick with us. Just so that you know, we will probably run about 10 minutes over our hour time frame. I apologize, but I'm going as quick as I can do it. So let's jump into cell respiration. So describe the process that allows organisms to use energy stored in biological molecules. So cell respiration now is the ultimate exergonic, catabolic, hydrolytic reaction. This is about pulling the energy back out of these molecules that we just created in photosynthesis. And remember, this is not just done by consumers. This is not just done by animals. This is also done by producers, plants. Even though plants go through photosynthesis and produce organic molecules, all organisms, animals, plants, every other form of life has to take those molecules and through cell respiration extract the energy to make ATP. So again, we are dealing with oxidation reduction reactions. In this case, we are going to oxidize glucose. If you look at the blue arrow, we're going to oxidize glucose, which means we're going to lose electrons and hydrogen and energy, and that's going to be oxidized into CO2. Where do the electrons, hydrogen, and energy end up at the end? That ends up in the reduction. We're going to take oxygen and reduce it into water, which we're ultimately going to see is the whole reason you have to breathe. If we put these side by side, we should realize that these are completely complementary reactions. If you look at the 2 blue arrows in both reactions, in photosynthesis we're reducing CO2 into sugar, but in respiration we're oxidizing sugar back into CO2. And if you look at the 2 red arrows, same idea, they're complementary. In photosynthesis, we're oxidizing water into oxygen, but in respiration, we're reducing oxygen back into water. The ultimate energy-coupled reactions. Photosynthesis being that key anabolic endergonic reaction, cell respiration being the key catabolic exergonic reaction. Now, in these reactions, we do involve electron carriers again. Now, in photosynthesis, we have this thing called NADP+. Here, we're going to use what's called NAD and FAD. We have NAD+ and FADH are the two electron carriers that are going to be used. These are oxidation reduction reactions. So what's going to happen is we're going to slowly oxidize the sugar in small steps. We have to control the energy flow. We can't take the sugar and oxidize it all at once, kaboom. You just killed the cell. You didn't sit in your car this morning and turn the key and ignite the entire gas tank at once. It wouldn't be pretty. Instead, you have a fuel pump which controls the flow of fuel from the tank to the engine, so you burn it in very small controlled doses. Well, in a sense, that's exactly what's going on in your body. We have to take glucose and not break it down in one big giant step. We have to break it down in multiple controlled steps. So that means we need some in-between molecules to help us transfer the energy as we're releasing it to the end of the process. And really, that's what NAD and FAD are going to do. They're going to pick up all these oxidized products that come out of glycolysis, oxidation of pyruvic acid in the Krebs cycle, and be the molecules that are temporarily reduced. So as we're oxidizing glucose, we're going to be reducing these energy carriers, NAD and FAD. NAD and FAD will eventually take all of their materials to the end product, or end step, called the electron transport chain, and then during the electron transport chain, we will oxidize the NAD and the FAD, re-release the electrons and the energy, and pass it off to oxygen to form water. Now that may not make a lot of sense right on the surface, so let's just step through it real quickly. So first step involves glycolysis. Glycolysis is followed by two steps, the pyruvate oxidation and what's called the citric acid cycle, or can also be known as the Krebs cycle. You'll hear me use interchangeably citric acid cycle and Krebs cycle. Krebs cycle after the gentleman who gets credit for discovering this cycle, Krebs. Some people consider pyruvate oxidation in the citric acid cycle one step. Other sources consider those as two separate steps. And then the final step is oxidative phosphorylation, which involves the electron transport chain and something called chemiosmosis. So we're going to take a look at each of these steps briefly. So glycolysis. Here's a simple picture of glycolysis. Basically what's doing in glycolysis is we're splitting glucose. We're taking this 6-carbon ring of glucose and we're going to break it down into 2 3-carbon molecules called pyruvate. And basically energy being released in several forms. During glycolysis, some of the energy is used to create ATP. You'll see on each side we create 2 ATP for a total of 4. But remember, to get the reaction started, it takes some activation energy, so we invested 2 ATP up front. So you have 4 ATP being produced, subtract 2 that it took to get it started. We have what we call a net gain of 2 ATP. Also, some of the energy has been invested in our electron carriers. As we've oxidized glucose, we had to reduce NAD to NADH. So some of the electrons and energy are being carried by the NADH molecules. And a lot of the energy is still in the 2 pyruvates. We've gone from a 6-carbon molecule to a 3-carbon molecule. They still have chemical bonds that were a part of the original sugar. So energy is currently in 3 locations. It's in the ATP, it's in the NADH, and it's in the pyruvate. And by the end of glycolysis, we have a whole net gain of 2 ATP. Now, glycolysis is a process that is carried out in the cytosol or the cytoplasm. It requires no oxygen. It requires no mitochondria. So we think this goes back to very early life forms, goes all the way back to prokaryotes, and was the primary way that energy was probably harvested. Well, if you're a prokaryote, if you're a bacteria cell, 2 ATPs, I can deal with that. 2 ATPs I can deal with. But if you're a human, 2 ATPs per sugar isn't going to work. It's not going to cut it. You're not getting enough bang for the buck. You got to get more energy than 2 ATPs. So glycolysis is going to happen whether oxygen's present or not, and whether you have a mitochondria or not. But for us large guys, we need more than 2 ATP. So there are two types of respiration that occur: aerobic, which is with oxygen, and anaerobic, which is without oxygen. Us large mammals, we need the aerobic respiration, so that's what we're going to look at first. So if oxygen is present and you have a mitochondria, we can go through the other steps. So from glycolysis, we go to pyruvate oxidation. The pyruvate actually can pass through a protein channel into the interior of the mitochondria, into what's called the matrix. And in here, we are going to oxidize the pyruvate into something called acetyl-CoA. So here's the 3-carbon pyruvate. Whoops. Right up here. And that's going to be converted into something called a 2-carbon acetyl-CoA. And in the process, you can see we're going to, again, reduce, because we're oxidizing something. You're oxidizing pyruvate, so something's got to be reduced. In comes our electron carrier. We're going to reduce NADH. And we're also going to produce some CO2. This chart sort of summarizes what's going to happen. The pyruvate, we're removing the carboxyl group at the top. That's going to be turned into carbon dioxide as a waste product. We are going to reduce NAD into NADH, which allows us to oxidize the pyruvate. And that's going to release CO2. And we're going to end up with a molecule called acetyl-CoA. Acetyl-CoA has the ability now to enter the citric acid or Krebs cycle. No more ATP produced here, but a little bit more NADH, which is holding on to energy, and a waste product of carbon dioxide. From there, we're going to flow into the Krebs cycle. So this shows the two connected. Here's our pyruvic acid. That's going to go through oxidation into acetyl-CoA. The acetyl-CoA is then going to be picked up by the citric acid cycle, where we will complete the breakdown of the acetyl-CoA into 2 carbon dioxides, make a little bit of ATP, and will produce more electron carriers that have been reduced. This is the one point where FAD does show up, and then NADH you can see is produced in greater quantity. So these two do connect together. By the time you're done with the Krebs cycle, you basically have completed the physical breakdown of the sugar. You've completed the physical breakdown of the sugar. So carbon dioxide is released. Now keep in mind, glucose produces 2 pyruvates, which means out of the 2 pyruvates you get 2 acetyl-CoAs. So for every glucose, you're going to take 2 turns of the citric acid cycle. So you can take all those numbers and double them if you want to know the quantities per glucose. These are being shown are the numbers per pyruvate or acetyl-CoA. So 2 carbon dioxides times 2 is 4, plus the 2 you released during the oxidation of pyruvate. There's your 6 carbons that were part of the sugar. And then you've got NADH, FADH2, and some ATP. The physical breakdown is complete, and we've gone through the first 3 stages, and I've got a whopping total of 4 ATP. Still not enough. I can't live on 4 ATP. I still don't have enough bang for the buck. So where's the energy? The sugar's gone now. We've gotten rid of the sugar. Where is the energy at? Well, the energy is being held by these carrier molecules. The NADH is holding on to the electrons, which means it's holding on to the energy that was in the bonds of that sugar, along with the hydrogens, and the FADH2 as well. So these carriers, that's where the energy is at. And so the final step is about taking those carrier molecules and extracting the energy back out of those molecules and turning it into ATP. So those NAD and FADH2s are going to show up at the electron transport chain, which is embedded on the inner folded membrane. So we've been in the matrix, Krebs cycles in the matrix, in the center, in the dense fluid. And now we're moving to the highly folded membrane called the cristae. And embedded in there are multiple electron transport chains. And you can see what's going to happen is the electron carrier's job is to deliver the electrons and hydrogen to the electron transport chain. So you're going to see NADH is going to be oxidized back into NAD+, which can travel back and repeat its job. FADH2 is going to do the same thing. It comes in a little further downstream, so it doesn't produce quite as much energy as NADH, but same idea, it's going to oxidize. We'll see later NADH can produce approximately 3 ATP per molecule, or FADH2 only has enough energy to produce about 2 ATP per molecule. So the key is the electron carriers come to the electron transport chain where they are going to be oxidized, releasing both their hydrogens and their electrons, and with the electrons goes the energy. Here's a bigger picture of the whole process. The whole thing is called oxidative phosphorylation. Oxidative because it requires oxygen to be the final electron acceptor, and phosphorylation because we're going to add a phosphate to ADP to form ATP. And there's two stages, the electron transport chain, which is going to create the hydrogen gradient, and chemiosmosis, which converts that hydrogen gradient into ATP, very similar to photosynthesis. So here's an enlargement of the electron transport chain. Here again is my NADH being oxidized into NAD+, releasing its electrons. Here's the FADH2 being oxidized back into FAD, releasing its electrons. And as the electrons flow through the electron transport chain, it is providing these pumps, these protein pumps, the necessary energy to pump hydrogen, in this case, out of the matrix into what's called the intermembrane space. And just like in photosynthesis, we're going to build up a proton gradient, a hydrogen gradient. We're going to accumulate hydrogen in this space between the outer and inner membrane. That again is going to build up that same concentration gradient, that same electrochemical gradient, and we're going to have this huge amount of pent up, built up potential energy that we want to tap into. And so the first step is all about building up that proton gradient. The second step, without the carriers delivering the electrons, we wouldn't have the energy necessary to build up that proton gradient. So the energy from the electrons is the energy being provided to the pumps to get that hydrogen gradient built. Now in the final step, just like in the chloroplast, we are going to provide a way for the hydrogen to finally get across the membrane. In this case, it's going to go from the intermembrane space back into the matrix. Again, we're going to use ATP synthase. And again, as these hydrogens flow, they in a sense go from a potential energy source to a kinetic energy source, and they allow the phosphorylation of ADP into ATP. Bingo, we have the desired product of cell respiration, ATP, which now can be distributed throughout that particular cell to do work, to do whatever work it is that that cell needs to do. However, we have to wrap up loose ends. Our loose end is right here. The electrons that went through the electron transport chain and were stripped of their energy are still here. The hydrogen that's getting pumped back across the membrane is still here. So we've got free hydrogen ions in the matrix. We've got free electrons, lower energy state, but free electrons. We've oxidized NADH and we've oxidized FADH2. Well, the rule is if you oxidize something, something's got to be reduced. And what's going to be reduced? This is where the oxygen comes in. Oxygen is what's going to get reduced, and the oxygen is going to be reduced. Sorry, I had something pop up there. The oxygen is going to be reduced into water. Oxygen will accept the two electrons. The negative charges of the electrons will attract the hydrogen, and we will form H2O, which is the final reduction. This, my friends, is why you breathe. This is why you need oxygen. If you don't have the oxygen, you can't complete this process. If you didn't need to do this, you might as well take your lungs out, put them on the table, you don't need them. If you don't have the oxygen, the whole system's going to back up. The whole system's going to back up because you can't oxidize the NADH and FADH2. You're going to run out of NAD and FAD. That's going to shut down glycolysis. That's going to shut down the Krebs cycle, and the whole process can't move forward. Now, almost there. I thank you for your patience. Here is an overview of the whole process. You can see we make 2 ATP net out of glycolysis. We get 2 ATP net out of the citric acid cycle. But the big payoff is when all those electron carriers, NAD and FAD, provide the materials for the electron transport chain to go through oxidative phosphorylation and produce a very large quantity of ATP. In this particular diagram, they claim 26 to 28 ATP. Other sources I've seen will say 30, 32, 34. That number varies on a daily basis. It varies from person to person. It varies based on your activity level, your diet. But you're going to produce a considerable amount of ATP in the oxidative phosphorylation. In total, this particular diagram says you'll produce about 30 to 32 ATP per glucose molecule. Others will claim somewhere around 34 to 36 to 38 ATP per sugar molecule. Either way, you're producing somewhere between 16 to 20 times the amount of ATP if you go through aerobic respiration than you would from just glycolysis. As a large mammal, I can live off of 30-some ATP per sugar. Finally, fermentation or anaerobic respiration. Some organisms live off of this. A lot of small organisms, prokaryotes, bacteria, this is their way of life. They're okay with 2 ATP. Could be they live in an environment with no oxygen. They don't have a choice. Could be like prokaryotes, they don't have a mitochondria. They don't have a choice. But they still need to go through some type of fermentation or anaerobic process because, like our cells, they have to regenerate the NAD. They don't use FAD. There's no FADH involved in fermentation. But like us, they have to regenerate the NADH back into NAD+, so they can continue to oxidize and reduce. So there's two main pathways that I'll share with you very briefly. One is called alcohol fermentation, and the other is called lactic acid fermentation. This is an example of alcohol fermentation. Humans have been using this for centuries. Here's the basic reaction. Here's glucose going through glycolysis, being turned into pyruvate. You're making your net gain of 2 ATP. You're oxidizing glucose to pyruvate. Therefore, you're reducing NAD to NADH. And if you stop at the pyruvate, that's where it ends. You're going to be stuck with NADH, which means eventually you're going to run out of NAD+. And if you run out of NAD+, there's nothing to reduce. And if there's nothing to reduce, you can't oxidize the glucose. And if you can't oxidize the glucose, you get no ATP. Death. So they've evolved the pathway where they can take pyruvate, convert it into acetaldehyde. You can then oxidize the NADH and reduce the acetaldehyde into ethanol. So we're going to take those hydrogens and those electrons, we're going to hand them off to the aldehyde right here. There's one of the hydrogens, there's the other hydrogen that were added, and we are going to reduce acetaldehyde into ethanol, alcohol. This happens when you ferment grapes to make wine. This happens when you ferment barley and hops to make beer, etc., etc. But it's a way to the whole key is, the whole reason we have to do this is to regenerate the NAD+, so we can continue to oxidize glucose, because without the ability to oxidize glucose, we can't produce the energy. And without NAD, there's nothing to reduce. Similar idea on lactic acid fermentation. Lactic acid fermentation, a little bit more direct. You're going to take the pyruvate and you're going to oxidize the NADH and reduce the pyruvate. You're going to add the hydrogens here and turn it into lactate or lactic acid. This will turn into lactic acid. One place we see this are the bacteria you find in yogurts. Yogurts often contain 5 or 6 different bacterial cultures. Yes, you're eating a live bacterial culture. And the lactic acid is what gives yogurt sort of its tanginess. The other key thing is that humans can actually do this step. This usually happens when you're under very strenuous exercise, and basically you are trying to produce ATP faster than your body can supply oxygen. Well, if you get to a point where you're producing ATP faster than your body can supply oxygen, this is a backup system that you can use temporarily. This is a backup system that you can use temporarily. And so what happens is you go into what's called oxygen debt, and the lactic acid is sort of an IOU. The more lactic acid you accumulate, it has negative consequences on muscles. Lactic acid causes muscle fatigue, it causes muscle soreness, it causes muscle cramping, all chemical signals to your body to, hey, slow down, stop what you're doing, we need to pay back our debt. Unfortunately, if you stop and you resupply the body with oxygen, you can reverse this reaction. You can take lactate and turn it back into pyruvate, and then you can go on with aerobic respiration. So keep in mind, these are all exclusion statements. I'm not going to read them to you. They're all saying you do not need to memorize specific steps and specific enzymes in the process. Understand what's going in, what's coming out. Photosynthesis, understand the light reactions, convert light energy into chemical ATP and NADPH, and then that is invested in the Calvin cycle to reduce carbon dioxide into organic molecules. Cell respiration, we're doing just the opposite. Last topic here is fitness, and what they're meaning here is on a molecular level, by having a variety of molecules, this often gives you a better ability to survive and reproduce. One classic example is plant pigments. There are different types of pigments in plant leaves. Chlorophyll, anthocyanins, carotenes are different types of pigments that are found in the leaf. And what that does is it gives the plant the ability to absorb light over a much larger spectrum, as this next picture shows. You can see chlorophyll A, chlorophyll B, and the carotenoids. They all absorb light in different parts of the spectrum. This is allowing the plant to gain more energy than it would if it only had one type of pigment. If it only had one type of pigment, it would not be able to absorb the same amount of energy, and therefore that would reduce the amount of photosynthesis. By having multiple pigments, multiple forms of the same types of molecules, it gives it an advantage, allows it to carry out much larger, more efficient levels of photosynthesis, giving it a likely chance to survive and reproduce. Other ones they talk about are phospholipids. There's actually 4 different types of phospholipids. They also talk about hemoglobins that you can investigate. Practice FRQs. I knew I was going to run into this situation. I've gone over time, and I knew I would never get to the practice part. So this is what I'm going to do. The lab experiences from Unit 3 are often parts of FRQs. In this section, a lot of teachers will have done an enzyme catalysis lab, a photosynthesis lab, and a cell respiration lab. I've listed some of the labs I do with my kids. You do not have to have done these specific labs. The AP question will never be on a specific lab, but on the concepts. Remember that 25-minute question, which makes up 2/3 of the AP score this year, is about interpreting and evaluating experimental results. If I had to predict, I'd say there's a high probability that could be based on a cell energetics problem. So what I'm going to do, because I'm out of time, is I'm going to post several practice FRQs based on Unit 3 that I've put together that will be designed to what I think the national exam will look like. I'll have those up by Tuesday. You'll see a link down below the video, and then I will make another short video and post a link there as well of me walking through the answers about a week later. So you can figure out how we can explore how these topics might be applied to this year's FRQs. I apologize for running over my time and keeping you longer. I appreciate you staying with us. So thank you for your participation. Stay safe, stay healthy out there, and enjoy the rest of your day.